Hello, today we're going to talk about DNA replication. I know the lighting is super bad, but I need to be in my study because I need my two computer screens. So we're just going to have to live with it. What you should have done so far in this lesson is you should have read the textbook, watched the Amoeba Sisters video, and downloaded a document that we're going to annotate today. First off, I just want to briefly talk about the textbook and a couple of things you've learned in the previous lessons regarding DNA structure. So the first thing here is looking at the cell cycle. So cells, like all living things, have a life cycle. And specifically when we talk about cells, we call it the cell cycle. And it's made up of three things. So it's made up of interphase, and then mitosis and cytokinesis. But we're only really focusing on this big stage today, which is interphase. And that's where a cell carries out most of its life. You can see that's why it's represented by such this large yellow section. And interphase is made up of G1, which is a stage of growth, as well as the cell carrying out its functions. We have this S phase, and then we have G2, which is another stage of growth. And again, it's also going to be carrying out its main functions. We care about specifically the S phase today in interphase. And S phase, as we can see, is DNA replication. And we need to learn how this process works. And our goal is to create, we start off with a parental DNA strand, and what we want to do is create an exact copy. And why? It's so when a cell replicates, that cell, those daughter cells, receive the exact same DNA as the parent cell. And that whole process of going from one cell to two is in lesson 5, but we specifically want to know how that DNA gets copied today in the S phase. Hopefully this is familiar from the textbook as well, and I got you to copy down this bit here, which says DNA replication is the process used by a parent cell to make an exact copy of its DNA. So for humans, in every single one of our cells, we have 46 DNA strands of 46 chromosomes, and each one of those DNA strands needs to copy themselves before they replicate. So DNA replication is essential for cell division, as both daughter cells need a complete set of genetic information. So that's so important, and that is the main function or the role of DNA replication. This diagram is also important because here you can see this is our original DNA molecule. Original DNA molecule. This is just one. In a human cell, we would have 46 of these. And what you can see is it splits, it replicates, and you can see that the two that are created both consist of one of the parental original strands, so one of these, and then they have the new complementary strand created. And if you read the textbook, we call that semi-conservative replication because each of the daughter molecules have one of the original parental strands. Finally, here we've got this picture just to represent this in a bit more detail and to remind you of the base pairs. So you can see if this is our DNA strand, we have that DNA strand breaking apart. And what's breaking apart? It's those weak hydrogen bonds between the bases. You can see as it's splitting, we've got new strands getting built. If we call this the parental DNA, well, can you see as it splits, the parental strands are what we call template strands or template strands, and the strands getting built are called the complementary strands. And then what we have is these two exact copies of DNA created. Now, it's important to remember that when these two get created, they still remain attached at the centromere. So if this is that original DNA strand here, just represented in a picture, when it creates its exact copy, it stays attached in the middle at a centromere. And that's very important when we start to look at when cells start to divide. It's still both considered one chromosome. Here we started with one chromosome. It's created an exact copy of itself, but it's still attached. So we still call this one chromosome. It's now just a double-stranded chromosome instead of a single-stranded. just a couple of reminders from the previous lessons as well that we need to know for a little bit later is first up, what is DNA made of? DNA is made of nucleotides. You can see here that it's just a whole bunch of nucleotides joined together. So a nucleotide is made up of a phosphate. If we're talking about DNA, a deoxyribose sugar, and a nitrogenous base, either A, T, C, or G. If we want to look at this in more of a molecular structure, we've got this diagram here. And again, you can see the nucleotides, phosphate, sugar, and base. And it's just a whole bunch of these joined together. And you can see here that the bases are joined together by hydrogen bonds. You can see here that between A and T, adenine and thymine, we actually only join, those only join by 2 hydrogen bonds just because of their structure, but cytosine and guanine actually join together by 3 hydrogen bonds because of their structure. But regardless, it's important to remember that these hydrogen bonds connecting these bases are weak and can be broken apart easily, and we'll look at that process soon. The final thing I want to point out that you should have learned in your last 2 lessons is that the 2 strands of DNA are anti-parallel. And what that means is they run in opposite directions. So we have here in this diagram the 5' to 3', and then the other strand is running 5' to 3'. So you can see they're running opposite to each other. We call that anti-parallel. Why is this called the 3' end? Well, if you look closely at the sugar, we can see the third sugar is free. And up here, if we look at this sugar, we can see the 5th sugar is the one that's free. Over here at this 3' end, the 3rd sugar is free. And down here at the 5' end, the 5th sugar is free. So it's just to do with the carbon, on that sugar and whether it's free or not. So pointing out that that's anti-parallel. Okay, I'm just going to pause this, pull up the worksheet that I asked you to download, and then we're going to go over it. Okay, so this is the worksheet that you should have downloaded. Now there's 2 options. If you want to print this out, just remember it's A3, so you might need to split it and print it separately. Shrinking to an A4, you wouldn't have enough room. But I've also provided you the coloured version, as well as if you feel like colouring in, there is a non-coloured version there as well. So that's what we're going to fill in today. With this worksheet, I'll just point out this little section at the top. So it says, note this diagram is half a replication bubble. So this diagram down here is half a replication bubble. Most textbooks will just show you this picture, and so did the diagram on the Amoeba Sisters video. Just wants to point out though that the same thing happens on the other side of the strand of DNA because we need to copy the whole thing. And just remember that the outside strands here, that's representing the original parent DNA. So that's the template strands, the ones in grey. And the black ones here in the middle are the strands getting formed, and they are your complementary strands. Okay, so as I go through this worksheet today, you're going to do the same thing and copy it down, whether you want to handwrite, whether you print it out, or you're just going to create textbook boxes and do the same thing. All right, so we're going to start at number 1 here, which is this orange enzyme, and that is DNA helicase. And we're representing this here by a zipper because what's its job? Its job is to unzip those 2 parental strands so that we can create the complementary strands as well. So I'll just pause. Okay, so DNA helicase is job. It's the enzyme that binds to the replication origin. And if we look at this picture here, you can see that replication origin is this section down here in the middle. So it binds to that replication origin on the DNA and begins unwinding that DNA helix by breaking the hydrogen bonds. So it breaks the hydrogen bonds between the bases. So if we look here at our DNA strand, okay, this is the parental DNA. You can see it's getting unwound. Okay, now just to point out that this helicase would have started back here and it's breaking the bonds between the middle, and it's heading in this direction. So it's heading in the direction I'm moving the screen. So as it comes through, it's breaking the bonds, and this helicase will continue to travel in this direction. So it's breaking the bonds, and these little outcroppy bits are meant to represent the bases. So it's breaking the bonds, the hydrogen bonds between the bases. And then as they break apart, you can see that replication fork is that Y shape where that breaking apart is occurring. There's two opposite forks extend away from the replication origin. So if you can see here, if we're looking at the whole DNA as a whole, we've got a replication fork happening here and a replication fork happening here. So what is the job of helicase? It is to unwind that DNA helix by breaking the hydrogen bonds between the complementary base pairs. And doing so, it creates a replication fork. So I'll get you to pause this video and copy that section onto your diagram. Okay, so now we're going to look at this enzyme that's sitting behind the DNA helicase. So you don't actually have to know the name of this. It's just that it's in the diagrams. I'll just point out what it is. And I mentioned it in the video. So it's this, I can't even say it, but topoisomerase, and it's a specific type DNA gyrase of this group of enzymes. And all it does is when that DNA helix is getting unwound by the helicase, it creates a lot of tension in the strand. So this, what is it called, DNA gyrase, it basically just breaks some of the bonds in the backbone, helps release some of that tension, and then builds them again. So it just stops the tension in the DNA molecule. Same with these guys, you don't need to know their name, but they're in the picture, so I thought I'd just mention them, and it's these SSBPs. You can see they're represented here, and basically their job is just prevent that strand forming back together because those hydrogen bonds want to reform. So it's just stopping that DNA molecule from closing back up before it can get copied. But really, we're going to focus firstly on this step one, which is the helicase unzipping. And now we're going to head off over to the other side of the page where we're going to look at this guy here, which is RNA primase. Okay, so we're looking at RNA primase now, which is this purple molecule. And you can see it's not only here, but it's also located here. And what it does, this RNA primase is building a primer. Okay, and you can see it, the prime is represented by this gray, and you can see that on this strand, we only have the one primer, but on this strand, we have multiple primers, and we're going to look at why that is the case as well. So let's just zoom in. So RNA primase is an enzyme that builds a short complementary segment of RNA. So it's actually building a segment of RNA. And what's the difference between DNA and RNA? It just means it's going to contain uracil at the base instead of thymine. But what it does is it builds a primer that is synthesized from, now this is important, so it says each RNA segment is called a primer and is synthesized from the 3' to 3' direction of the template. So you can see we're starting with the 3', so it's built in this direction. But it says, hence, the segment itself, because it's anti-parallel, is built in a 5' to 3' direction. Now, your syllabus statement requires you to know the direction that the enzymes are getting, the strands are getting built. So you need to know that the template strand, the primer gets built from the template strand from a 3' to 5', but the primer itself is built from a 5' to 3'. So that's the direction the complementary strand is going. It says primase, so the purple guy here, is needed because the enzyme DNA polymerase responsible for laying down the bases, so the guy that actually puts down all the bases, needs the primer because the RNA primase provides a jumping off point. So this guy, the DNA polymerase, doesn't know where to start. It needs the primase to build this primer, and then that's the jumping off point for the DNA polymerase to know where to start building. So again, pause the video and copy down this, and then we'll start with the next guy. So we've started to build our complementary strand. We've popped a primer in. Now we're going to get the DNA polymerase to finish building that strand. So I'll just point out, this is where the helicase would have started, and it would have traveled in this direction. As it's come through, RNA primase has come here and built a primer, and then that's going to be the indication where DNA polymerase can start building the complementary strand. That is why we have this little guy here with one of those starting flags in a race, because it's representing the starting for the rest of the starting for DNA polymerase to start building the complementary strand. If we look at the little picture we've put here for DNA polymerase, now if you don't know that's Bob the Builder, then anyway, so this is Bob the Builder, so he's a builder. And so that's what DNA polymerase is. He is a builder and he's building our new complementary strand. This is like RNA primase. This enzyme can only add DNA molecules in the 3' to 5' direction of the template strand. So 3' is building this direction. But if we're looking specifically at the complementary strand, it's building it from a 5' to 3' direction. And you need to be super clear when explaining this on your external exam, because it's a syllabus statement, so you can bet it's going to appear somewhere. It says DNA polymerase adds nucleotides starting at the end of the primers because it needs that to know when to start. The complementary strand is synthesized by adding complementary nucleotide units according to the sequence of the template strand. So you can see that these little random guys here, these are nucleotides, so they consist of a phosphate, a sugar, and a base. Now if this up here on the template strand was an A, then the DNA polymerase would put a T here. Okay, so it's building a complementary strand opposite, so A's with T's and C's with G's, to that of the template strand. Okay, so I just want to come back out to the big picture. Now, number 6, like I said, these guys are nucleotides, so they're just free in the nucleus, and the DNA polymerase is using those to create that complementary strand. Now you'll notice that when we look at this template strand, we've built our primer, we're just happily going along and building our strand. And remember, we're building it on the complementary strand from a 5' to 3' direction. This looks super nice and neat, but down the bottom here, it looks a bit messy. And that's what we're going to talk about now. 7 and 8 are leading and lagging strand. So we're just going to talk about this neat strand first because it's the easier one. And then we're going to figure out what is happening here and why does this look different. And it's to do with the 5' to 3' direction that polymerase can only travel through on the complementary strand. Okay, so the leading strand, like I said, is this one here. Why is it called the leading strand? So it says this is the newly synthesized. Synthesized means to copied or made. So this is the newly synthesized DNA strand that is built in the same direction as the replication fork. So like I said, the helicase would have started here and is traveling this direction, and we have this following that. It says is built continuously in one long segment and requires only one primer. So here you can see we've only got one primer, as well as it only requires one DNA polymerase and one ligase, which we'll talk about later. It says only one DNA polymerase is needed for the leading strand, as the direction of DNA synthesis is the same direction as the movement. So DNA synthesis is going in the same direction as helicase, which on the template strand is 3' to 5'. On the complementary strand is 5' to 3'. Okay, so now we're going to look at the lagging strand and why it looks a little bit different. Okay, so the lagging strand, you can see here that we're heading to the same direction as the helicase, so this direction. And here you can see as helicase is traveling this way, our lagging strand is getting built this way. And so we want to look at why that is. So it says this is the newly synthesized DNA strand that is built in the opposite direction of the replication fork. So as the fork is going this way, we're building it this way. It is built using short fragments of DNA, and these are called Okazaki fragments. So how can I move my screen? If you can see here, we've got our primer getting built. So like I said, we're building this direction. So we've got our RNA primase building a primer here and here and here. And then following this, we've got DNA polymerase building these pieces of complementary strand. So each little piece here is going to get called an Okazaki fragment. So the lagging strand is built discontinuously in fragments away from the direction of the replication fork. So since DNA polymerase can only add DNA fragments in a 5' to 3' direction of the template strand, it is required that DNA be built away from the direction of the fork. So let's just say that we've started with helicase over here, it's coming through. So then what happens is as it's coming through, primase starts a section on the direction of the 3' to 5', the direction it can travel on the template. And then we've got the DNA polymerase coming in and building that section. As the fork frees up some more, another primer, DNA primase, is going to build a primer here, and then the polymerase is going to come in. As the fork keeps traveling this direction, we're going to build a new section. So we just keep building these sections of both the primer and the complementary strand. So we get these chunks called Okazaki fragments. So it's called a lagging strand because it's lagging, it takes more time. So our next job is to look at, well, we've got all these broken pieces of strands, how are we going to join them together? And that's where our next enzyme comes into play. So you'll notice I've skipped number 9 for a moment. I just want to look at the enzyme that joins these Okazaki fragments together. So it says the enzyme that joins the unattached sections of DNA by forming bonds between them. So it's going to form bonds between each of the broken segments. It says ligase is needed to attach together the Okazaki fragments. DNA ligase is needed many times on the lagging strand and only once on the leading strand. Okay, so when we've got this section up here built in those two sections, then that would get used once, but we've got to use it a bunch of times when we're building that lagging strand. So you can see here it's represented here, and we've got a picture of a glue here to represent it's joining those Okazaki fragments. The last enzyme that we're going to look at, we're not going to give it its specific name, but because there's a few of them, but we're going to look at this yellow guy here. So we've got written here other enzymes. So there's other enzymes that are in play here. We're just looking at the 4 main runners. So we're looking at our DNA helicase and our RNA primase, which is our purple guy here, the DNA polymerase, and the DNA ligase. But there are other enzymes. And so it says DNA polymerase, remember it's the builder, the guy that's building our complementary strands, says they rarely add the wrong nucleotides, but sometimes instead of matching an A with a T, it might match an A with a C. So it says if it does happen, there are actually proofreading and repair enzymes that correct this error. So there are enzymes that go back and check DNA polymerase's job. It says mutations will only occur if this backup system fails, but we do have that backup system, which is great. So that's looking at all of the enzymes and their role. So we just want to look at what's our end product now. So we're just going to look at number 11. So the final thing we just want to reiterate is that DNA replication is what we call semi-conservative. So replication results in 2 DNA molecules. Each one consists of the original parent strand as well as the complementary strand. And so therefore, because we've kept some of the original in each of those new strands, we call it semi-conservative. So that is our process of DNA replication. Make sure you've got it filled in. You do need to know the role of those 4 enzymes as well as the direction that they can work. So whether it's the 5' to 3', the 3' to 5' of the template, or the 5' to 3' of the complementary strand. So it's important that you know the roles of each one and the direction that they can move. So hopefully that helps. And make sure, just for homework, that you read this and try and learn it. And then we're going to do some review stuff next lesson with some textbook questions. All right, send me any questions if you have them.